Professor Davis here, and we are going to now take a closer look at viruses, viroids, and purons. Now, this was talked about a little bit early on in some of our videos. Remember that viruses are not living, and so because of this, they are known as being acellular. They do not have a cell. And so in order to be a living thing, you have to be made of at least one cell. So these guys aren't. So these are known as non-living particles. One thing that's neat too is the word virus is actually Latin, and it's Latin for poison. So again, it's to show that it causes illness or sickness. Now, the reason these guys cause a lot of times these illnesses and sicknesses is because they are obligate intracellular parasites, meaning they have to gain access inside of the cells in order to reproduce. They can't reproduce without it. And so what they do a lot of times is they're going to hijack the machinery of your cells in order to make more viruses. All right, and so this is what makes them obligate intracellular parasites. So let's look at some basic viral structures. So when we look at viruses, they do contain an inner nucleic acid core. This core is going to be made up of either DNA or RNA. They do not have both. Prokaryotic and eukaryotic cells do have DNA and RNA. Viruses do not. They have one or the other. They are going to store or hide away this DNA or RNA in a protein capsid. This outer protein capsid a lot of times is going to offer protection for that DNA and RNA. And they come in all kinds of different shapes. Now, sometimes viruses have an envelope. So just like how an envelope helps hide or conceal a letter, you know it's in there, but you don't know what it says. You can't tell because the envelope hides it. The envelope can help hide a virus from things like your immune system. So a lot of times animal viruses will have envelopes which help them evade your immune system. But an envelope is optional. It's not always present. So let's do a really quick comparison of bacteria versus viruses. We talked about bacteria already in a prokaryotic video, and so now let's do a little bit of a comparison. So we're going to look at several things. We're going to look at cellular organization, metabolic activity, how does it respond, how does it replicate, does it mutate, just different things like that. All right, so let's take a look here. So the first thing we want to look at is the cellular organization. So bacteria does have cellular organization, but viruses do not. Because remember, they're acellular. They do not have a cell. Metabolic activity, bacteria does metabolism. They do chemical reactions, okay, but viruses do not. Response, bacteria responds to its environment. Viruses do not. Replicate, yeah, they both can replicate. However, bacteria replicates through binary fission, whereas viruses can only replicate within host cells. So it's the only time they can replicate. Mutates. Bacteria can mutate, and so can viruses. Viruses actually can mutate very quickly, which makes it difficult to develop a lot of times effective vaccines against them, or at least certain groups of them, like the flu, things like that. Now, let's look at some structures. So, do viruses contain a plasma membrane? Well, a plasma membrane is a cell structure. So, bacteria has it, but viruses do not. Organelles or ribosomes. Bacteria have ribosomes, but viruses do not. DNA and RNA? Yes, bacteria has both. However, remember, viruses either have one or the other. They don't have both. So the answer here is no. It's either or. They have DNA or they have RNA. Bacteria, is it filterable? Okay, is it size dependent? No, it's not. But viruses are. Okay, they are very small, and so they are filterable. We do see, are they sensitive to antibiotics? No, bacteria is, but we cannot use antibiotics against viruses. Viruses, however, they are sensitive to a chemical that your body releases from your cells, which are called interferons, right? So, but antibiotics do not touch viruses. So let's look at some of the general morphology or shapes that we see with viruses. All right, so some viruses are known as helical viruses. Helical viruses are seen here in C, okay? So C is a helical virus. We have some viruses that are considered polyhedral. Polyhedral means they have lots of sides to them. This is like adenovirus and poliovirus. Now, these are seen here. Okay, so they have all these sides of their capsid and they are polyhedral. Others are enveloped. So enveloped viruses, they can be enveloped helical viruses or they could be enveloped polyhedral. Enveloped helical are example of like the influenza or flu virus, whereas enveloped polyhedral is like the herpes virus. All right, and so here's an example. We then see that we can have, so the first one here is the enveloped helical, D is enveloped helical, and B is the enveloped polyhedral. Now, complex viruses are present. Some of these complex viruses are things like the pox virus, okay, as you can see here, and also the bacteriophage. So they're going to be a little more complicated. Bacteriophages, guys, look pretty scary. They're the ones that remind me from like Toy Story, where it's the little machine legs with the doll's head on top as it comes out from underneath the Sid's bed. That's what this guy kind of looks like. But one thing unique about this is, as scary as a bacteriophage looks, it does not affect us. Bacteriophages, they are viruses that infect bacteria. So what's weird about viruses is they will infect anything and everything, from bacteria all the way up to us as humans. They can also infect plants, other animals, protists. They can infect almost any living organism out there. So since viruses are not living, their taxonomy or the way they're grouped is different. So these guys get grouped based on the type of nucleic acid. Are they a DNA virus or are they an RNA virus? They also a lot of times get grouped by their morphology, their shape. Are they helical or are they polyhedral, like with all the sides? Do they have an envelope? Yes or no? Okay, that could also put them into a category. And their host range. Who do they infect? Do they only infect humans? Do they infect humans and say like pigs, or do they affect humans and monkeys? Their host range is important. That's like their niche. So the genera name suffix for a virus is going to be virus. Okay, so when we look at this, we'll have something in front of it and then virus will be at the end that lets you know that it is a virus. So example here is norovirus, denotes the viral genera. So that's what tells you it's a virus. The family name suffix could be the Viridae. Okay, so when we look at their family name Viridae, that's what denotes a viral family. All right, and so that would be multiple types of viruses underneath that one group. All right, so how do we cultivate viruses in a lab, specifically like bacteriophages? How can we get them to grow in a lab? How can we get them to where we can study them more? Well, we can use what we call the plaque method. So the plaque method is where you would take a nutrient agar and you would create a bacterial spread plate. Now, spread plates are talked about in some of our labs, but we would take the bacteria and we would cover the whole plate with it. So that allows it to grow into what we call a lawn. You then will add a thin layer of liquefied agar with bacteriophages in it. So the agar will have bacteriophages and you'll put that on top of your lawn of bacteria. Now remember, viruses have to get into bacteria in order to replicate. And in that process, when they replicate, they destroy the bacteria. And so if they're present and they do like the bacteria that's also growing there, you will see that they create these little clearings. These clearings are known as plaques. So we have a plaque forming unit, or a PFU, and it's these areas where you can actually see through the agar. These like kind of looks like holes. There's no bacteria present there because the viruses have cleared them out. Okay, the viruses have killed them off, making more viruses. All right, so that's one way, and this is going to be more for bacteriophages. Well, what about animal viruses? Animal viruses, we can actually cultivate them using living animals. Okay, so we can use living animals. A lot of times they might use like the foot pads of certain animals or things like that. They can also use eggs. Okay, so the eggs have the embryos inside and they can grow the viruses there. And we can also use cell cultures. These cell cultures are going to look for what we call cytopathic effects, where they are CPEs. And so you can see that it affects the line of the cells if the viruses are present. All right, so these are some different ways to cultivate or to grow viruses in the lab setting. Another thing we can do when we're looking at viruses is we can do what we call a serological ID of them. And this helps us be able to identify specific viruses. And this is where we have the virus, which normally has an antigen present, like something on it, on its coat, on its envelope that does not belong. And your immune system recognizes it does not belong. And so because of this, there are certain antibodies that can be against that virus. If the virus is present, the antibody will attach to it. And if it attaches to it, then you actually get a reaction. All right, and so if you look here at these examples, you can see A has no virus, and so there's no reaction, okay? B has the virus, but no antibodies, and so we see that it's going to have this issue where the blood is going to be affected by the virus, whether it's being destroyed, and so all of it spreads out. So we have a different reaction. But on C, we see that the viruses with the antibodies help hold on to the virus, so the virus cannot affect the blood cells, and we see that it is a different kind of reaction. It's back to that no reaction state. All right, and so this is why antibodies from your immune system are so important to help fight viruses. And a lot of times what's used here is called the Western blot method in the lab. All right, so how do these viruses do multiplication? How do they make more of them? Even though they have to get inside of your cell, how do they do this? Well, first they have to be able to invade the host cell. They got to get inside. If they can't get inside, then they cannot do this process. So this means once they get inside, they commandeer the host metabolic activities. They take over. Okay, it's like they're holding the cell hostage, and they're like, you're going to do what I want you to do. And a single virion, a single virus, is capable of producing thousands once it hijacks a cell. All right, so this is a big deal. Now, there are two ways that a virus can multiply. The first is called the lytic cycle. The lytic cycle has 4 steps, or technically 5 steps to it. So the first step is attachment. Attachment is that it has to attach to the cell. All right, so here the bacteriophage is attaching to the surface of the cell, picture 1. Picture 2, we have penetration. Penetration is where the viral DNA or RNA enters the cell. Now, the whole virus does not get in, all right? That capsid, that stuff stays out. Just the DNA or RNA needs to be inside the cell. Once it is, we see step 3. Step 3 is biosynthesis. This is where the DNA is going to replicate, and they're going to make proteins using the ribosomes of that cell to make more of the components of the protein capsids. Then in step 4, maturation, they're going to put all the parts together. They're going to put the replicated DNA inside the new capsids, and they're going to get ready to be released. They're maturing. And then step 5 is lysis. This is where the cell bursts, releasing all the new viruses that are made. Okay, so this is called the lytic cycle. The next one is the lysogenic cycle. Now, the lysogenic cycle has the same steps as the lytic cycle, but it has one addition. This one addition is called integration. This is where once the DNA gets inside of the cell, that DNA integrates into the bacteria or cell's DNA. So you notice here that turquoise DNA from the virus is going to incorporate into the pink DNA. And it's going to sit there. And as these cells divide, each new cell that divides is going to have a copy of this virus. So this creates an issue. If one virus can make 10 in the lytic cycle, we are now increasing our odds of how many viruses are going to be made with the lysogenic. Because one cell divides into 2, 2 divides into 4, 4 divides into 8, and so on. And every one of those cells has the virus DNA in it. It's being copied too. And once it gets triggered and that viral DNA comes out and it makes the components, instead of one cell making 10, I now have 100 cells making 10. It's a huge increase in the number of viruses that are released. All right, and so this creates a potential issue. Right? And this is why some of these can stay dormant for a long time and they flare up occasionally, like with cold sores like herpes virus, or this is why too, you can get infected with HIV. It gets inside your cells. We can't detect it. And this is why you have to be tested over a year's period of time. If you think you've been exposed, you're not HIV negative until after you've had a year's worth of tests. If it then still comes back negative, we see that you didn't have the virus. All right, so this makes it a little more dangerous when we look at viruses. Okay, so let's take a look at when we put the two kind of stages together. So if you look here, you have the lysogenic cycle shown at the top, and then you have the lytic cycle is this bottom part. So the lysogenic cycle does contain the steps of the lytic, but it has that integration piece first. So the lysogenic cycle is longer, whereas the lytic cycle is faster. Okay, now there are lots of different clips on comparison of the lysogenic cycle and the lytic cycle and how they work on YouTube or other areas that you can look online. You should look at those and see how they actually work and how they overlap. All right, but you can take some time to look at that. So here's an example with the actual lysogenic cycle, and this one specifically is for a retrovirus. A retrovirus is like the virus of HIV. So if you'll notice here in the picture, it attaches. So attachment occurs and then penetration happens. In this particular case, because it has an envelope, we do see that it's going to attach and then it's going to get kind of uncoated and unwrapped. Once it's done this, it is RNA. This is an RNA virus. So we have to work backwards. That's where the retro comes in. So RNA is going to be turned into DNA. The DNA is going to then be integrated. This process of going from RNA to DNA is called reverse transcriptase. There's an enzyme that's going to do it. It's called reverse transcription, and the enzyme is reverse transcriptase. Then the DNA is going to integrate into the host DNA. And you can see how the yellow DNA integrates into the blue inside the nucleus. Once conditions become right, this biosynthesis of the RNA and its proteins then start to happen. They mature and then they release. Now, one thing that's unique here is because this is an enveloped virus, when they release, they bud off with a piece of your now cell membrane. So they can hide from your immune system because their cell membrane looks just like yours. Their coat looks just like yours. So they're able to hide and evade your immune system. And this creates a bigger issue. So retroviruses are here. Again, on YouTube, there are plenty of videos that show this process with the retrovirus replication. So take a look at those. All right, so we have a comparison here in this chart where it's a bacteriophage and viral multiplication is compared. So the bacteriophage is the virus that affects the bacteria, whereas an animal virus is the one that affects, like, say, us. All right, and so when you notice, a bacteriophage does not go through the process of integrating. Okay, it's going to not need to be uncoated. There's no envelope to it, and it's going to get released relatively quickly. Whereas animal viruses are a little more complex because they contain that envelope. They have to actually be uncoated. We do see a lot of times they are going to integrate into the DNA, which means they enter in that lysogenic cycle, and so they are going to be a little more complex than the bacteriophages. So let's talk a little bit about viruses and cancer. All right, so transduction is the transfer of DNA from one cell to another, and this is done by bacteriophages. So bacteriophages can introduce in new bacteria. This may result in cellular transformations where the cells actually change. So conversion of a proto-oncogene to an oncogene can occur here. Now, a proto-oncogene is a pre-cancerous cell. Or gene. It's not cancer yet, but if it mutates to an oncogene, it is now a cancer gene. And a lot of times when we have viral infections and we have all these different issues that we're exposed to with bacteriophages and things like that, it can trigger these genes to change to where they go from precancer to cancer. Okay. We also see that they can mutate a certain area in the DNA that's called the tumor suppressor gene. This gene normally makes sure that you don't create tumors, but if it's mutated, tumors can also begin to form. Now, a normal cell can be changed to an abnormal cell, and it can be done by adding in a piece of DNA from a virus. DNA oncogenic viruses are things like HPV, okay, human papillomavirus. This can cause cervical cancer, EB, which is Epstein-Barr virus, and hepatitis B. RNA oncogenic viruses, ones that potentially cause cancer through RNA, is going to be the T-cell leukemias and lymphomas. They're going to be viruses that ultimately cause leukemia and lymphoma. So here's a chart here that is showing you if you are going to be infected with a virus, kind of what happens. In that red area, you see you're infected, but you don't have symptoms until the active infection ramps up where the viral load gets very large. So the number of virions or viral load is actually really high, and so it goes up on that chart. Now, eventually your immune system takes care of it and it drops back down. And a lot of different viruses, that's the end of the infection. At the end of the red, it's done, you're good. However, some viruses follow what we see here with the green and the blue, okay? So the green is a persistent infection. This is one that stays around. So you have a pretty high infection and you have a lot of symptoms at first, then it drops back down and then it stays steady. Now, it continually goes up and it stays steady for a while till it starts to actually bridge up again. These are things like HPV, HIV, and HBV. All right, so these are types of viruses that we would consider persistent infections. On the other hand, you have the blue, which are the latent infections. So you'll notice that the red, it goes down and it stops. But later, months later, years later, it ramps back up on the blue side. These are viruses that are present in your body once you've been exposed, and they stay. But they only come out when the opportunity is good. So when your immune system is down or when you're stressed, you will have an outbreak or an infection with these again. And this includes cold sores, which are caused by the herpes simplex 1, genital herpes, which are the herpes simplex 2, and shingles, which is also a herpes virus, and it's the zoster, okay? And it's linked to the chickenpox virus. All right, and so these are latent. They sit and they wait, and they can come back. Okay, and so this is giving you an example of the different types of infections. All right, so some terms that are important here are going to be things like virions. Virions can actually infect animals, plants, and bacteria. They are the complete virus. So when we talk about a virus in the virion, it's talking about the complete virus, the protein capsid, the nucleic acid core. If it's a viroid, viroids are seen in plants, and these are infectious RNA particles. We don't see these in animals or bacteria, but we do see them in plants, and they're called viroids. Another one that is found in animals are called the purons. Purons are pretty scary because they are infectious particles that are protein infectious particles. These are where proteins get mutated and changed, and then they peer pressure your proteins to mutate and change. And this is a big deal because a lot of these are actually found in the brain, okay? And they cause issues. So like in sheep, we call it scrapie. In cows, we call it mad cow disease. And you've probably heard of this, and it makes them go crazy. Like it affects their brain and kind of makes them go crazy. And with that, we can't eat that meat because that meat has been infected. All right, and so they got to make sure that it's not spread. And so if a facility has cows that are infected with mad cow disease, they've got to destroy all those cows, and they cannot be used for food, okay, because it could then affect humans' brains if they were to consume it. And this is what happens when we talk about some human diseases that comes along with Kruger and Creutzfeldt-Jacobs disease. These are purons of mutated proteins, and they were linked where we found them in cannibals, cultures that do cannibalism. You're thinking, who still does cannibalism? It's not like when we talk about, there are groups that are very remote that if you come in as an outsider, they might eat you and that sort of thing. I'm not talking about that. These guys are normally where the cannibalism is related to their religion. And so in order for their loved ones when they pass to go to the next life, whatever it might be in their mind, they normally have to consume the heart and the brain of that family member. Well, the brain is where this is located. And so they give themselves a death sentence with this. But as modern medicine comes about, it's hard to convince them that they don't need to do that, because then they feel like they're letting down their loved ones. All right. And so this is a battle that they're kind of fighting with this, with certain diseases like you see here. Now, last but not least, guys, viral diseases, you're going to learn them and what causes them for your final exam in my microbiology class. The ones you need to know are listed in your disease sheets on study sheet 4. Study sheets 1, 2, and 3 focus on the bacteria, whereas study sheet 4 is going to focus in on the viruses. So make sure you're studying that for your final exam. If you have any other questions, please let me know.